Hello and welcome to the Chapter 13 lecture on genetics and molecular biology. This lecture is going to help you study for the final in Plant Science 711. All right, first let's take a look at transposons. We talked before about how DNA can break and reform readily. And this is basically how transposons work. They're also known as jumping genes. So here we can see a multicolored ear of corn, and we can see that one of the kernels has the purple pigment from the anthocyanin, but not in all of the cells. The cells that are not producing the anthocyanin in that kernel of corn actually have the gene interrupted by transposons. So when the transposons jump in, they can disrupt the function of a gene that is going to essentially break the gene, or when they move around, they can then restore the function of the gene when it goes back to its normal form. Okay, so this actually can be used to look at the function of a gene when the genes are dissociated or turned off. Then we can see what the actual function of the gene is. And these were initially discovered by Barbara McClintock, who was working on maize in the two types of molecules that make up DNA, or the chromosomes, I should say, are DNA and protein. So the DNA is going to have three parts. There is the nitrogenous base, or the nucleotide, the 5-carbon sugar, which in this case for DNA is deoxyribose. That's why DNA is called deoxyribonucleic acid. And then the phosphate group. So the information is contained in the nitrogenous bases, and then you'll have the alternating sugar and phosphate groups that are going to get the structure to the DNA. Okay, so there are 4 types of nucleotides, or 4 letters of the genetic code. Okay, we have the purines, which are adenine and guanine. Adenine represented usually with an A, guanine with a G. And here we have 2 aromatic rings that are bound together for the purines. On the other hand, we have the pyrimidines. Those are made up of cytosine and thymine. Cytosine usually represented with a C, and thymine is represented with a T. So this base is just going to be a single ring for the pyrimidines. Okay, so we have the nucleotides that come together, and they're actually hydrogen bonded together. So you can see that here we have the purine bound with the pyrimidine, and then you see the dotted lines between them, and that is showing the hydrogen bond. Okay, and then these are in the form of, the 2 strands of the DNA are in the form of a double helix. And the backbone here is made up of alternating sugar and phosphate groups. Okay, as I mentioned before, the purines will bond with the pyrimidine. So example, you would have G with C and A with T, which we can also see right here in the middle. Okay, so the nitrogenous bases, or the part that has the genetic code, is going to be the nucleotides. So not all of the DNA codes for a gene or for a protein or a trait that we can see. Some of the DNA is still not well understood or thought to be potentially nonsense. And there's some DNA sequences that are involved in the regulation of the gene. Okay. And then the genome, that's going to comprise the sum total of all of the organism's DNA. Now, during the S phase, or the synthesis phase of the cell cycle, that's when the DNA is going to be replicated. So the steps in this, the double helix is going to become unzipped or separated. And then each of those strands is a template for the new strands to be laid down. Okay, the enzyme that is responsible for DNA replication is DNA polymerase. Okay, and the precise sequence of the template, or that is to say the complementary sequence to the template, is going to be laid down going in the opposite direction. Okay, so each of the DNA molecules in that case has one strand from the original strand, and then one new strand that was used, having the original strand as a template and replicated by a DNA polymerase. So for that reason, it's called semi-conservative replication, since half of the molecule was conserved and half of it was laid down fresh. Okay, so here we can see the example. Here's the original molecule with the double helix, and it has been unzipped. And now we have new nucleotides that are being laid down and giving the complementary sequence that bonds with the original sequence. Okay, so now moving on from DNA and starting to get into RNA. RNA is going to come into play when the genes are actually expressed. So remember, the DNA is the template. It has all of the genetic information, but there's differential expression of genes in different environmental conditions or different parts of the plant's life cycle. For example, we talked about germination versus flowering. They're going to be different genes that are expressed. Okay, so the first thing is going to be that DNA is transcribed to RNA, and then RNA will be translated to protein. Okay, so this is sometimes referred to as the central dogma of molecular biology. And just to run over this again, it's DNA, RNA, protein. And the processes that are occurring here is that the DNA is transcribed to RNA, and then the RNA is translated into protein. Okay, so in terms of transcription, I mentioned that it has to do with RNA, right? So the messenger RNA is going to be what consists of the transcripts of the genes that are later going to be translated to proteins. There's also a transfer RNA or tRNA that's going to be responsible for translation. It'll bond to the mRNA. And then ribosomal RNA that's associated with the ribosomes, it's also part of the machinery for translation and production of protein. That makes sense since we know that ribosomes are the factories of proteins within the cell. Okay, so RNA, this is interesting because we have 4 nucleotides that are going to make up RNA, but slightly different. So instead of A, T, C, G, we're going to have adenine, cytosine, guanine, and uracil. So we'll have U instead of T. The other difference here is that we will also have a single-stranded molecule for RNA. So RNA is more of a transient type of molecule. It's going to degrade rapidly, whereas DNA is very stable. And if you think about the functions of these, it makes sense because DNA is the template. We want that to be around all of the time. But since RNA is part of our differential expression, it's going to degrade rapidly so that the cells are able to respond to the environmental stimulus pretty quickly and able to change gears if needed. Okay, so only the portions of the genome that are going to code for genes are actually transcribed into RNA. The rest is typically called non-coding DNA. Okay, so how does this work? There is a promoter region that's upstream or at the very beginning before the gene or the coding sequence. And that promoter region is going to recruit the transcription factors that need to bind to the site. So the promoters are basically saying, come to the DNA party. Okay, now at the end of the coding sequence is a terminator DNA sequence. At that point, the transcription enzyme, which is the RNA polymerase, is going to detach from the molecule. So we have a clear sign of where the beginning is and where the end is. All right, so a lot of the non-protein coding DNA is responsible for gene regulation. Okay, so how does the transcription get carried out then? So the tRNA, remember, is transfer RNA and it's part of the building of the protein. It's going to be responsible for translating our codons that are made up of 3 nucleotides. So that's going to be 3 of our nitrogenous bases. And each of those codons codes for 1 amino acid. Remember that the amino acids are the building blocks of protein. Okay, so the tRNA will have one end that's bonded to the messenger RNA, and then the other end will bond to the specific amino acid that's indicated by the codon in the messenger RNA. Okay, so every amino acid is going to require at least 1 transfer RNA, and there'll be a specific anticodon loop that will match and it'll recognize and bond with the codon in the messenger RNA. Okay, there are also some ribosomal RNA that are going to be transcribed in the nucleus, and those are used to make the ribosomes themselves, and that is going to be part of the translation machinery in order to make proteins. Okay, so as I mentioned, for translation, the messenger RNA is going to consist of the transcript. It's and for a coding region of the DNA that codes for proteins. And the codons consist of 3 nucleotides. The interesting thing here, and it took a while for researchers to figure out, is that there are 3 nucleotides that consist of a single codon, and there are only 20 amino acids. It turns out that there are 64 possible combinations of the codons, and they only code for 20 amino acids. So there is a little bit of backup that is built into that. So if there's potentially an error in the code, it could possibly not actually affect the genetic code or the protein that's produced, because there's more than 1 codon that codes for the different amino acids. Okay, and then this A, T, C, and G is pretty much universal, and we can see it in all of the different kingdoms. Okay, so here we can see a diagram or a cartoon with our example of what happens during translation. So first we have the tRNA or the transfer RNA. It will have an anticodon loop that will recognize the codon in the messenger RNA, and that will be for every amino acid. Okay, and this is going to be signaled by a free ribosome that's going to occur in the cytoplasm that will bind to the mRNA. Okay, and then the codon that's going to set the start frame or set the reading frame is going to be AUG. All right, and here we can see it again. We have our DNA, which is going to be coiled up in the chromosomes of the nucleus. The DNA will be transcribed to RNA. Here we can see messenger RNA consists of the transcript, and then that messenger RNA will be translated into protein. Okay, so the transcription occurring in the cytoplasm or in the nucleus, I should say, and then the translation is occurring in the cytoplasm. Okay, so mutations, these are possible way for changes to occur in the DNA sequence. If you remember when we talked about chapter 12, I mentioned that the process of meiosis is going to give us new genetic combinations, but it's not possible to get completely new genes by that process. Okay, so mutations, though, are a possibility, and they're caused by mutagens. The mutagens are going to be agents that generate mutations, just as the name implies. Some possibilities are UV, okay, ionizing radiation such as gamma radiation, or chemicals such as ethyl methane sulfonate. That is a mutagen. Okay, there are natural repair processes within our cell that will detect these errors and then correct them. Okay, now there's two different possibilities here. Okay, a somatic mutation, taking this back to plants, it's going to occur in a body cell. So you might think of potentially a mutation in a leaf that's going to change the leaf color, for example. Okay, so unless that leaf is vegetatively propagated or cloned, then that mutation is not going to be carried on to a future generation. However, the other possibility is to have a germline mutation, which would affect the reproductive cells. Now that would be a heritable mutation that's going to be carried on to future generations. Okay, so within molecular biology is the field of cytogenetics. And here they're studying the different chromosome behavior and the ways that chromosome structure can be changed and what the effects are. One of the possibilities here is inversion. This is going to be where a piece of the chromosome actually flips around and it's going to bind to the same chromosome, but it will have a different orientation. So the end of the chromosome would now be located somewhere in the inner part of the chromosome. Okay, so when we have this occur, we don't see crossing over in meiosis because they don't pair well with the homologous chromosome, the other sister chromatid, and for that reason, they're going to be inherited in blocks. Another possibility other than inversion is going to be translocation. So here we have a section of the chromosome that is going to break off and it will bond to another chromosome. Okay, so you might have something from chromosome 3 that breaks off and then bonds to chromosome 4. And these are important in speciation. They're especially well known for grasses. And I thought this was pretty interesting. I found this article showing that the chromosomes from wheat, rye, and barley can actually bond with each other. And they are actually going to associate and move together in meiosis. Okay, so for plants, a nucleoid, which is an unusual or irregular number of chromosomes or an irregular shape of chromosomes, which could be caused by the translocation or inversion, can actually help to contribute to the fitness of the plant. It can be responsible for speciation, whereas in humans, for example, it can lead to deleterious traits. Another thing that we want to think about here is the polyploids. The polyploids are going to have many copies of the genetic information, whereas our diploid individuals had 2 copies. We might have 4, 6, or even 8 copies of the genetic information. And the polyploids often will have advantages because they'll have higher yields. Some examples here that we have are cotton, peanuts, strawberry, and also sugarcane. Okay, so let's get into Mendels Peas here. So during the 1860s, we're going to look at the first set of traits, which is the tall and short pea plant, or the plant height. Okay, when the parental generation was crossed together, the resulting generation was the F1. All of the resulting offspring were tall, and they were 100% identical to one another. Okay, now when the F1 generation was intercrossed, then there was a specific ratio that was observed. There were 3 tall individuals and 1 short individual. So we had these 3 tall individuals that were exhibiting the dominant trait, and then 1 short individual that in fact exhibited the recessive trait. Okay, then F2 generation is the subsequent generation that follows when the F1 plants are crossed together. Okay, so here we can see a little cartoon giving us an example of what this might look like. We have the two parents, the tall parent and the dwarf parent. When the parental generation is crossed, then we get the F1, and here we get all tall. So we only see the dominant phenotype here. Okay, but in the F2 generation, we start to see the segregation of traits, and now there are 3 tall and 1 dwarf individual. So our phenotypic ratio here is 3 tall to 1 dwarf. Okay, so what was behind all of this? The first is the law of unit characters. So here, the law states that there are factors, they always occur in pairs, and those are going to control the inheritance of various characteristics. So remember, Mendel was working with peas, so that was a diploid organism. They had 2 sets of the genetic information, or 2 sets of chromosomes. And although the term alleles had not been coined yet, here we're interested in the 2 different forms that specifically code for a trait of interest. We were looking at height here. Okay, and then the homologous chromosomes are going to have genes at the same position or the same locus. Okay, so if I look at, chromosome 3 in an individual, there are going to be 2 sets in the pea plants. Okay, they might have different alleles or different forms of that particular gene, but that gene is going to be found in the same position, whether it's on one chromosome or the other. Okay, then there's also the law of dominance. So we may have a dominant trait, which in this case was the tall trait, that will mask the expression of the recessive trait. So if the dominant allele is present, even if it's just 1 copy, it'll mask the expression of the recessive trait. All right, and then what about the difference between phenotype and genotype? The phenotype, that's going to be something that you can physically see or measure, whereas the genotype is asking, what is the genetic code of this individual? So the phenotype is going to be influenced by both the genotype and the environment. When we talk about Mendel's model and we say, what is the genotype, then that would be either homozygous or heterozygous for that locus. So if we have a homozygous individual, then both alleles are going to be identical. That means they have both the same forms of that gene. Okay, for the heterozygous individual, the alleles are going to be different from one another. So we might have, for example, for the homozygous individual, big T, and for the heterozygous individual, big T, little t. Okay, so let's take a look at this in a little bit more detail. This is the same cross. We have our, we have green and yellow pods, okay, but very similar to the one that we looked at for height. Okay, we're gonna start with 2 true breeding parents. That means that the parents are going to be homozygous. So the first parent is homozygous for green pods. The possible gametes from that individual are going to be just big G, because it only carries the dominant allele. And then the genotype of this parent is big G, big G. So it's all big G. Now the second parent was homozygous for the yellow trait. So it has the alleles little g, little g. The only gametes that I can contribute to the next generation are going to be little g, because this is a homozygous yellow individual. Okay, so when the parents are crossed, then you get the F1 generation, which is all green. The F1 generation is all identical. And the reason why we see all green is because the dominant allele is going to mask the expression of the recessive allele. That is to say, the F1 generation is all heterozygous. So we have big G, little g on both of the individuals for their genotype. Now, the gametes that these individuals from the F1 generation can contribute are going to be either big G or little g. Since they have both types, they can create gametes or reproductive germ cells that are going to give one type or the other. Okay, so that's why we see the segregation of traits in the F2 generation. And we wind up with our phenotypic ratio of 3 green and 1 yellow. Well, here we can set up our Punnett square. So you have 4 cells that represent the offspring, and then you put the gametes around the edges. So we have the big G and the little g, because you can see this first individual from the F1 generation has big G and little g. Those are the possible gametes. And then we get the same gametes from the second F1 individual. Since it is identical to the first, we have big G and little g. What are the resulting offspring from this? Well, when we cross big G with big G, we're going to get homozygous dominant, or big G for this individual, which of course exhibits the dominant phenotype of the green pod color. When we cross the little g here with the big G, we get the big G, little g. That is to say, it's the heterozygous individual. The genotype here, big G, little g. This one exhibits the phenotype of the dominant trait, okay, because the big G is going to mask the expression of the recessive trait represented by the little g. So in other words, we don't see the yellow pods as long as there's at least one copy of the dominant allele present here. Okay, same thing here for the third one, big G and little g together. We see green pods, heterozygous genotype, and then 25% of the time, or 1 out of 4, we're going to get our recessive, homozygous recessive individual. That is to say, with 2 copies of the recessive allele. In this case, little g, exhibiting the recessive trait. So the recessive trait is only going to be actually expressed, or we're only going to be able to see it if the dominant allele is not present at all. Okay. And for this model, which is our monohybrid cross, because we're just looking at one trait here, we're looking at the pod color. Then we have a 1 to 2 to 1 genotypic ratio. So remember, we have 1 homozygous dominant individual, 2 heterozygotes, and then 1 homozygous recessive individual. And then what we can actually see without knowing the genotype, we know we have 3 green and 1 yellow. That is our 3 to 1 phenotypic ratio. Okay, so now with the dihybrid cross, it gets a little bit more complicated here. We're going to look at pure breeding parents that differ by 2 traits now. So they would otherwise be identical except for these 2 traits. Now, the law of independent assortment comes into play. So we have 2 different factors, or as they're now called in modern genetics, genes, that are controlling these 2 traits on the chromosome. They're going to independently assort. Okay, so remember in meiosis, when we have the crossing over, the genes for 2 different traits are going to assort independently. So we might have an individual with the green pods that could come out either tall or short, depending on the pollen grain that unites with the ovule. Okay, or we might have a yellow pod individual that comes out tall or short. So there are different possible combinations. Now, if the genes are very close together on the chromosome, or that is to say they're linked, they're located very near to one another, then they do not segregate independently. This is just like shuffling the deck. The cards that are located close together in the deck are going to be likely to travel together when the deck is shuffled, and they're associated with each other because they're so near together. Okay, that would be like the linked genes. The unlinked genes are going to be located far away from each other, or here they say on different chromosomes. So those are going to be subject to the law of independent assortment. Okay, so for our dihybrid cross, then we're going to get our F1 generation that's all identical, but it'll have dihybrids. So that means it'll have heterozygotes at both of the loci of interest, since we're looking at 2 traits now instead of just 1. So in that case, it can produce 4 different kinds of gametes. Okay, we can use our Punnett square to figure out what the genotypes are and the expected phenotypes. And for dihybrid cross, we're going to see a different phenotypic ratio. Okay, so we're going to see 16 individuals total here as a result of this instead of 4. We have 2 traits, so therefore it is 4 squared or 16 individuals. The ratio 9 to 3 to 3 to 1. Let's take a look at that in a little bit more detail. Okay, so first of all here, we'll start out with our homozygous dominant parent having the green pods and the tall height. The possible gametes from this individual are going to be the dominant allele for green pods and then the dominant allele for tall plant. Okay, that's going to be crossed with a parent that differs at those 2 loci. So we'll have the homozygous recessive genotype for pod color, which is going to give us yellow pods, and then the homozygous recessive genotype for height, which will give us the dwarf individual. Okay, so when these parental individuals are crossed together, we're going to get the F1 generation. Here we'll have heterozygotes. So the phenotypes that are reflected in the F1 are going to be the dominant phenotypes. So these are essentially just going to resemble the dominant parent for those 2 traits. So we have the heterozygote, big G, big T, little t. It shows us our dominant phenotype. But here, this is interesting because it has both big G and little g and big T and little t. We're going to see these 4 different combinations of the gametes. So we have the all dominant, we have our green potted but short individual, we have a yellow potted tall individual, and then we have one that resembles our yellow potted dwarf. Okay, remember these are the gametes, so it's only going to be half of the equation because we're going to get 1 copy from mom and 1 copy from dad. Okay, that's just what happens when the F1 gametes are crossed together. Okay, so remember the F1s are going to be identical. Okay, and then you can see where it gets a little bit more interesting in terms of the combinations here. So when the F1 are intercrossed, then we're going to see all of these different combinations again. So remember the F1 are identical to each other in their genotype, so we'll have the same thing in terms of the gametes from each parent. So we'll have the green potted tall combination here. We have a green potted short combination, a yellow potted tall combination, and then a yellow potted short combination. Okay, so we're going to wind up with some different combinations here from those. The first one, we have the all homozygous dominant. So this is going to resemble our homozygous dominant parent that has the green pods and is tall in height. Okay, here we have in this example homozygous dominant at the first locus and then heterozygous at the second locus. So here we get green potted and tall again, even though we have a slightly different genotype at the second locus, it's just going to look dominant. Okay, looking at the third individual here, we have the heterozygous combination at the first locus and then homozygous dominant at the second locus. So still green and tall, not really seeing too much variation yet. Okay, for this next combination at the end of the top row, we have heterozygous at both loci. So again, we're going to see the dominant phenotype green and tall. Okay, moving right along to the second row from this cross, we will get homozygous dominant at the first locus and heterozygous at the second locus, green, potted, and tall. All right, the next one, we have homozygous dominant at the first locus and homozygous recessive at the second locus. So here we get to see something that we didn't see in the parental generation, and you could see perhaps where this is actually useful for breeding because we have new combinations. All right, here we have a green potted short individual. Okay, moving along to the next one, we have all heterozygous at both loci. The next one, we have heterozygous at the first locus and then homozygous recessive for the second locus. So we have a short green potted individual again. All right, moving along to the third row here. This combination gives us heterozygote at the first locus, homozygous dominant at the second locus. So we have green potted tall individual. Okay. Next, we have heterozygous at the first and second locus. So green potted and tall once more. Okay, moving along. Here we have the homozygous recessive for podcolar, and then at the second locus, we have homozygous dominant. So we start to see some new variation, new combinations that we didn't see in our parentals or in our F1. Here we have a tall individual with yellow pods. Okay. Next, we have the combination for homozygous recessive at the first locus and heterozygous at the second locus, again giving us the yellow potted tall individual. Okay, going down to our last four in the bottom row. Okay, so this combination here gives us heterozygous at the first locus and heterozygous at the second locus. Here we have green potted and tall. All right. The next one will give us heterozygous at the first locus and homozygous recessive at the second locus. So we have a short green potted individual, one of our new types here. And then for the next one, we have homozygous recessive at the first locus and heterozygous at the second locus for height. So giving us a yellow potted tall individual. Finally, just 1 out of 16 times, we get our individual with all homozygous recessive, heterozygous for, or I should say homozygous for color and for height. So we have the yellow potted short individual. Okay, let's take a look at the back cross in more detail. Okay, so for our back cross, we're going to start with 2 true breeding parents, and they're going to be different at our gene of interest. Okay, so let's imagine that our all right, so let us imagine that we have 2 different varieties of basil. And we have a variety that's excellent, it has excellent aroma and excellent potency, and it has purple flowers, but it happens to be susceptible to powdery mildew. So this could be a variety that is very well liked, but it needs to have some disease resistance incorporated or introgressed from another parent. Okay, so that parent that has most of the traits that we want is going to be the recipient parent, okay, as opposed to the donor parent, which is going to be the one that is going to give the trait that you're trying to introgress. Okay, so remember when we cross these 2 individuals, then we're going to get the F1 generation, okay? And each parent is going to contribute half. Okay, so we'll wind up having 50% of the genetic information coming from parent 1 and 50% from parent 2. And remember that these are also going to be all identical to one another. Okay, so for our first cross, or BC1, we're going to cross P1 with our F1 generation. Okay, the result of this cross is going to be the BCF2. Okay, and if we cross our P1 and our F1 together, then we're going to have 75% of the genetic information coming from our recipient parent, which was P1. Remember, P1 had all the traits that we like, but it didn't have the disease resistance. Okay, so for our BC2, or our second back cross, then we're going to cross our P1 again with our BCF2. Okay, and here we're not going to see that all of the individuals are identical. Okay, this will give us our BCF3, or third filial generation of our back cross, and that is going to have 87.5% of the genetic information from P1. So it's going to fairly well resemble P1 at this point after our BC2. Okay, and then for our third back cross, it will go as such. Okay, so we have our P1, which is our recurrent parent or our recipient parent, being crossed with our BCF3, and that's going to give you BCF4. Okay, you could continue in this fashion. Okay, but here we're seeing about 95% similarity to our recurrent parent or our recipient parent that had all of the desirable traits. And presumably we could find an individual in there that is going to exhibit our trait of interest from the donor parent. Okay, I have covered the test cross in the previous lecture on genetics in some detail. All right, so other than Mendelian inheritance, where we're going to see our 3:1 or 9:3 to 3:1 phenotypic ratio, there are also some other possibilities. So one of those would be incomplete dominance or a lack of dominance. So here, if we have a heterozygous genotype, we're going to see a combination of the two phenotypes. Okay, so this would be like if you had a red flower and a white flowered individual, those were crossed together, and the result was a pink flower. That would be incomplete dominance. Okay, we can also have pleiotropy or interaction between genes, where more than one gene is responsible for the production of a protein, and these genes may code for proteins that react or work together in order to produce the products that actually give you the trait that you would see for the phenotype. Okay, so the genotype is going to say which form of the allele or which form of the gene is present. In some cases, if you have a defective enzyme or an enzyme that doesn't work or is not produced properly, okay, then that is what's going to give you your recessive phenotype. Okay, so sometimes that is because of a broken enzyme. Now, in terms of quantitative traits, those are going to be traits that occur over a range rather than just having discrete outcomes. So a good example of this would be for height or for yield, whereas a discrete trait would be something like flower color, where you only have two possibilities, like white or purple. Okay, days to flowering is also going to occur over a range of values. So you're not expecting to see all of the individuals to flower at the same time. And what is interesting is that because the environment influences this, it can account for some of the range as well. Okay, so the area of the chromosome that's associated with the quantitative traits can be mapped using statistics to identify the QTLs or the quantitative trait loci, and that'll basically tell you where on the genetic map that trait is located. And it's like a model for the chromosome. Okay, we also have extranuclear DNA. This was discussed in chapter 3. You have the mitochondrial and the chloroplast genomes. Okay, and if you recall, we also discussed endosymbiotic hypothesis, or in endosymbiont theory. The idea being that the mitochondria and the chloroplasts were prokaryotes or single-celled organisms like bacteria that were then engulfed by a larger organism like a protist, and that gave rise to the eukaryotes. Now, it's true that the DNA sequences of the mitochondria and the chloroplasts are similar to bacteria, so there is some evidence to support this. Another thing that's interesting here is that the mitochondrial and the chloroplast genomes follow maternal inheritance. So a plant trait that is related to this would be the seed coat. So the seed coat is maternal and it's going to be passed along maternal lines. Okay, so remember the linked genes are close to one another on a chromosome. And because they're located with one another, they're also usually associated with one another and inherited together. Okay, so the crossing over is more likely to occur between genes that are far apart on a chromosome rather than two genes that are very close together. So if you have traits that are associated with one another, that could also be called a linkage block. Now, where the crossing over has occurred, those would be the recombinant types. Remember, that's where we get our new combinations of alleles. Okay, you can actually do crosses to find out how often recombination occurs for the traits of interest and use that to map the chromosomes. So one map unit or one Morgan is going to be the same as 1% crossing over between the pair of genes. Because remember, the closer the genes are to one another, the more likely they are to cross over. Okay, so we'd expect them to then cross over with a higher frequency since it's more likely. Okay. There's also Hardy-Weinberg theory, and this says that the proportion of dominant to recessive alleles in a large enough population with random mating is going to remain static from one generation to another, unless there is some selective pressure. And we also see that this doesn't hold true in small populations. Okay, so we've covered all of these topics. Thank you for joining me this afternoon.